Okay, let's unpack this. Imagine you're handed, like, a bag of Lego bricks. But instead of the usual instruction manual, you're just told, build something that can open a specific lock. The precise shape, right, the exact three-dimensional arrangement of those bricks becomes absolutely critical for success. This is actually a bit like the microscopic world of molecules. Their exact shape, their conformation, isn't just some minor detail. It dictates everything they do. Whether a drug binds effectively to a protein in your body, or maybe how a new material gets its unique properties. And the profound challenge lies here. Predicting these precise 3D shapes, especially for larger, more complex molecules, well, it's historically been incredibly difficult and computationally really expensive. Traditional methods feel a bit like trying every single Lego combination until you just stumble upon the right one. It takes forever. But here's where it gets exciting. Cutting-edge generative machine learning models are now stepping in. They offer a much more direct, targeted, and frankly intelligent approach.
Yeah, what's truly fascinating here is how powerful AI, specifically models known as diffusion transformers, or DiTs, which you might be familiar with from their incredible ability to generate stunning images, even videos, how they're being reimagined for something completely different. They're diving into the microscopic world of molecular structures. So today, we're taking a deep dive into some groundbreaking new research. It tackles the unique challenges of adapting these powerful tools to the, well, continuous irregular geometries of molecules, and it showcases some truly ingenious solutions. Our mission, really, for this deep dive is to explore the innovative architectural elements and the experimental findings that make this kind of revolutionary adaptation possible and effective. It gives us a peek into the future of molecular science.
Okay, so let's really clarify the core problem we're talking about. When we say molecular conformer, what do we mean exactly? We're talking about the precise three-dimensional arrangement of atoms within a molecule. Think of your hand, maybe. It's always your hand, right? But it can take on many different shapes, a fist, an open palm, gripping something. Each of those is kind of like a different conformer. And why does this matter so profoundly in the real world? Because that exact 3D shape is the key. It's the key to a molecule's biological activity and its physical properties. Yeah, this understanding is absolutely fundamental to fields like computational drug discovery. Imagine a drug needing to fit perfectly into a protein, like a specific key into a specific lock. Or think about material design, where a material's strength or conductivity or flexibility fundamentally depends on how its atoms are arranged in 3D space. Now, traditionally, methods like molecular dynamics, MD, and Markov chain Monte Carlo, MCMC, they try to explore this incredibly vast conformational space. They do this by simulating physical movements or probabilistic sampling, but the core limitation is kind of inherent. They're just computationally expensive. They require so many simulation steps just to move from one plausible conformer to another, let alone trying to search all the possibilities exhaustively. It's like wandering through a massive maze hoping to find all the hidden treasures.
Exactly. And this raises a really critical question for, scientific acceleration. How can we bypass these lengthy step-by-step simulations and find those treasures more directly? This is where generative machine learning models offer a paradigm shift, a really big one. They don't simulate step-by-step. Instead, they learn the underlying distribution of all plausible conformations directly from data. It's like having an AI guide who, instead of exploring every single path in that maze, has already learned a map of where all the high-value conformers are likely to be. And it can directly sample from those promising locations. So the broad concept is that these generative models, they learn an unknown data distribution. Once they've grasped it, they can then create diverse high-quality samples from that distribution. And we've seen the incredible success of diffusion transformers, these DiTs, in other fields, particularly, generating highly realistic images and videos. This sets the perfect stage for this paper's core contribution, taking that generative power and applying it right down at the fundamental molecular level.
Okay, here's where it gets really interesting for me. The brilliant team behind this new work decided to take that highly successful diffusion transformer architecture and, well, fundamentally adapt it for molecules. Just think about the profound difference for a moment. Images are these neat grid-structured arrangements of pixels, rows and columns. Molecules, though, they're irregular, continuous 3D point clouds. Basically a collection of atoms floating in space, connected by bonds. That's a massive conceptual and technical leap, from pixels to positions in 3D space. So their model, which they've aptly named DiTMC diffusion transformer for molecular conformer sampling, it's framed around predicting the 3D arrangement of atoms, their precise Cartesian coordinates. But critically, it's conditioned on the molecular graph, where atoms are the nodes and chemical bonds are the edges. It's sort of like giving the AI the chemical blueprint, right? The abstract connections, and then asking it to figure out the exact physical construction and the stable way those connections arrange themselves.
Precisely. And to achieve this really complex transformation, they've introduced several innovative technical aspects that are truly at the forefront of AI and chemistry. First, let's talk about conditional flow matching, or CFM. This isn't just, another diffusion process. It's the highly efficient generative backbone that learns a time-dependent vector field. Picture it as learning a precise, continuous set of instructions or flow lines. These lines can smoothly transform samples from a very simple, easy-to-generate prior distribution like just a cloud of random points into the much more complex, structured, and specific molecular data distribution. A key academic advantage here is CFM's remarkable flexibility. It allows transformations between arbitrary distributions, which is crucial. It doesn't force assumptions about the initial state, unlike some older methods that often rely on a simple, maybe overly simple, Gaussian prior. The underlying mathematical framework involves what they call stochastic interpolance, and the model essentially learns to predict the clean sample directly. This is key for efficient and accurate generation. It's like teaching the AI not just to connect the dots, but to draw the perfect curve between any 2 points, no matter how complex.
Okay, so CFM is kind of the elegant mathematical engine driving this. It guides the AI from random noise to a clear molecular structure. But even with that powerful engine, the AI still needs to truly understand the terrain of molecules, which, like you said, is incredibly bumpy and irregular compared to a neat image grid. How do they sculpt the DiTMC architecture itself to really navigate and interpret that molecular landscape? That sounds like a whole different set of clever design choices were needed.
It absolutely is. And yes, within the DiTMC architecture, there are indeed some very clever architectural innovations. One major area revolves around their conditioning strategies. See, each DIT block, as it processes information, needs to receive crucial conditioning signals, not just the current time step in the generation process or just info about individual atoms, but also pairwise information derived from that molecular graph we mentioned. Now, the core innovation here, and this is really neat, lies in their all-pair conditioning. So instead of just considering atoms directly bonded to each other, which is a common simplification you see in many models that use graph geodesics, this basically means finding the shortest chemical path between every single atom pair in the molecule, no matter how far apart they are structurally. And this is profoundly powerful. It allows the model to capture global structural information, things like long-range relationships, the overall molecular connectivity, stuff that simple bond pair conditioning often completely misses. You can actually see the impact in the results. If you only rely on, say, node-only conditioning, the model can sometimes output chemically invalid structures, maybe stretched bonds or even atom swapping places. But with node and all pair conditioning, it fundamentally preserves the bonding patterns and the chemical structure, which is absolutely crucial for the fidelity and, importantly, the chemical validity of the generated molecules. It's like giving the AI a truly holistic, detailed blueprint, not just a list of who's immediately next door.
Wow, okay. So they're not just looking at direct connections, but understanding the full, intricate structural relationship between every atom pair. That makes total sense for building truly realistic molecular geometry. But when we talk about understanding shapes, molecules also have these inherent symmetries, right? Like a molecule doesn't change its identity just because you rotate it in space. How did they tackle that specific fundamental challenge to make sure the model isn't just mimicking shapes, but truly grasping these forms?
Yeah, that's a great point, and it leads us directly to another major innovative contribution. This one builds fundamental physics directly into the AI. Positional embeddings, PEs, and Euclidean symmetries. The challenge, exactly as you highlighted, is handling these inherent symmetries in molecular data. A molecule is the same molecule whether you rotate it, translate it, whatever. You really want the AI to inherently recognize this, rather than treating a rotated version as something completely new. So they explored a hierarchy of 3 types of PEs. First, absolute PEs, APE. These are simple. They just encode the raw Cartesian coordinates and don't enforce any symmetry. They're basically a baseline. Then there are relative PEs, RPE. These use displacement vectors between atoms. This improves translational invariance, meaning the model knows it's the same molecule even if it's shifted, but crucially it doesn't account for rotation. The real academic breakthrough, and where the ingenuity really shines, is their Euclidean positional embedding, or PE3. This isn't just some minor technical tweak, it's a fundamental architectural shift. It constructs what are called SO3 equivariant pairwise PEs using something called spherical harmonics. Now, without getting lost in the deep mathematics, What this achieves is profound. It builds in knowledge about how molecules behave under rotation and other transformations. It respects the full set of Euclidean symmetries relevant to molecular geometry. This ensures the model inherently understands that a rotated molecule is still the same molecule. It's almost like embedding a tiny physicist right into the AI's brain, telling it, hey, that's the same thing, just viewed from a different angle. And crucially, to ensure these symmetries are preserved throughout the entire network, not just at the input, they designed an SO3 equivariant self-attention mechanism. This means that every token, every piece of information flowing through the network transforms equivariantly. It's a highly sophisticated way of processing information. It guarantees the model's outputs are always chemically sound and respect the fundamental physics of molecular interactions. It uses advanced concepts from group theory, things like irreducible representations and Clebsch-Gordan tensor products, to ensure the entire operation is symmetry preserving. This is absolutely crucial for maintaining accuracy and chemical validity from the beginning to the end of the generation process. This foundational innovation lets the AI learn a level of chemical intuition that purely data-driven models, well, they simply cannot achieve.
Okay, wow. So they've effectively given the AI a deep understanding of molecular physics, not just like a huge data set to memorize. That feels like a massive leap forward. But what is all this innovative architectural work, all this clever embedding of physics? What does it actually mean for the DiTMC model's ability to generate accurate molecular conformers? Does it truly pay off in practice?
It absolutely does pay off, and the experimental results are pretty compelling. Models incorporating full Euclidean symmetry with this PE3 approach demonstrate significantly improved sample fidelity. If you look at their detailed data, especially in figures like 2A and 2B in the paper, the PE3 variant consistently outperforms other versions, particularly at low RMSD thresholds. What that means is it generates geometries that very closely match the ground truth structures. We're talking near-perfect molecular shapes, essentially. And the reason for this superior performance becomes clearer when you examine the generation process itself, like in figure 2C. Models without these equivariant PEs tend to accumulate higher errors as they get closer to the final data distribution, near T1. This leads to noisier, less accurate structures at the end. Equivariance, in stark contrast, maintains accuracy throughout the entire generation trajectory. It ensures the structures are precise and chemically valid at every step. It really demonstrates the power of building these fundamental principles directly into the AI's design. Now, having said that, we do need to acknowledge the computational trade-offs. There's always a trade-off, right? The benefit of higher fidelity, while transformative, it does come at a cost. The equivariant PE3 model is approximately 3.5 times slower during training, and it's about 3 times slower during inference compared to its non-equivariant counterparts, like the ones using APE or RPE, even when they have similar parameter counts. So this is an important practical consideration for researchers for drug discovery companies. You have to weigh the need for the absolute highest accuracy against computational efficiency, depending on your specific application and, well, your resources.
Right. So you gain incredible precision, that near-perfect shape, but it demands more computational muscle. That feels like a classic dilemma in high-tech development. But let's dig a bit deeper here. Is getting a perfectly shaped molecule enough, or does it also need to act like a real molecule? Are we just talking about geometrically similar structures, or are these generated conformers truly chemically viable? Do they have the right properties?
That's a crucial distinction, absolutely. And a real testament to the model's success is that it goes beyond just geometric metrics like RMSD. It also accurately predicts critical ensemble properties, things that are vital for chemical validity, like a molecule's energy, its dipole moment, the HOMO-LUMO gap. This is incredibly significant because it indicates the physical validity and the chemical meaningfulness of the generated structures. They aren't just pretty shapes, they actually behave as real molecules would. In fact, this model outperforms other leading methods in predicting these properties, especially for energy prediction, where it can be up to a factor of 4 better. This truly underscores the depth of the AI's understanding. It's moving beyond mere geometry to capture actual chemical behavior. Furthermore, the model shows really impressive generalization performance. It can effectively generalize to larger and previously unseen molecules. They showed this on the GeoMXL dataset. And what's even more remarkable, perhaps, is that it performs on par with, or sometimes even better than, the best prior methods out there, like a model called MCSL, while using significantly fewer parameters. That's a huge win for efficiency and scalability. It means this approach could potentially be applied to more complex real-world problems in the future without needing an exponential increase in computational resources. And finally, their ablation studies, where they systematically removed parts of the model to see what breaks, provided critical insights into their design choices. They definitively confirmed that all-pair conditioning, the one using graph geodesics for every atom pair, is superior to simpler node-only or bond pair conditioning. This really reinforces the importance of capturing that global structural information we talked about. They also showed how careful architectural choices, like avoiding something called index positional encoding, are essential for preserving desirable properties like permutation equivariance. This ensures the model isn't just memorizing some arbitrary atom order, but truly understanding the underlying chemical structure itself.
Wow. Okay, this deep dive has really opened my eyes to how AI is pushing the fundamental boundaries of science. It's genuinely mind-bending, isn't it? To think that the same kind of AI technology that's generating photorealistic images can now sculpt the molecule down to its atomic coordinates. What's truly astonishing here isn't just that it works, but how precisely it seems to adhere to the underlying physics. That's not something you'd necessarily expect from, say, an image generator. It's important to note, though, that this work, like a lot of cutting-edge research, is currently focused on small and medium-sized molecules, and relies on having high-quality ground truth conformers for training. But, these are common addressable challenges when you're developing revolutionary new methods like this.
Exactly. And if we connect this back to the bigger picture, what this work really presents is a powerful, principled framework for molecular conformer generation. It achieves this by cleverly leveraging the inherent symmetries and the graph structures of molecular data within these cutting-edge generative models. It's really a testament to how fundamental innovations in AI architecture, especially when they're deeply informed by scientific principles, can lead to truly transformative breakthroughs. Breakthroughs in fields like drug discovery and material science, potentially accelerating the pace of discovery pretty significantly.
So what's next for this frontier then? This deep dive really shows us that by understanding and, crucially, building in the very nature of molecules, their symmetries, their complex graph structures, AI can achieve a level of precision and maybe chemical intuition that's truly exciting. It definitely raises an important question for the future. How will advancements in AI, especially those deeply rooted in fundamental scientific principles and the specific physics of the domains they operate in, how will they continue to reshape the landscape of discovery in chemistry, biology, and probably far beyond? You've been listening to The Deep Dive. We really hope this exploration gave you some truly eye-opening aha moments, and perhaps a new appreciation for the hidden, intricate shapes of the molecular world.